Now, the very first way by which these antimicrobials can inhibit protein synthesis is, like, we know that tRNA, it will come and it will attach to A site only. But if we can give a drug which will inhibit this attachment of tRNA on A side, means if tRNA do not attach to A side, then proteins cannot be synthesized. Okay, so this action is stopped by a drug, that is tetracycline. Okay, so what tetracycline does, it will inhibit the attachment of tRNA on A side. Okay, so we know tetracycline, it is an antimicrobial. We know that it is a protein synthesis inhibitor. But what exactly tetracycline does? Tetracycline will inhibit the attachment of tRNA on the A site. Next step of protein synthesis in bacteria was formation of peptide bond. Okay, so we know that these two amino acid, they will bind to form a bond that is known as peptide bond. So we can give a drug that will inhibit the formation of peptide bond, and if a drug inhibit formation of peptide bond, it will also inhibit the protein synthesis. Okay, so we have got a drug known as chloramphenicol. The main mechanism of action of chloramphenicol, yes, it is also protein synthesis inhibitor, but the main thing that chloramphenicol does is it will inhibit the peptide bond formation. Next, third step in protein synthesis of bacteria was to produce translocation. Okay, so we know that once this peptide bond is formed, then this ribosome will be moving, which is known as translocation, forming a new A site, isn't it? So this is translocation. But if translocation does not occur, protein synthesis will be inhibited, right? So we have got drugs that will inhibit translocation, and these drugs are macrolides, clindamycin, and quinpristine. Okay, so three drugs we have got, or three group of drugs we have got, that will produce action, that is inhibition of translocation, by which they will inhibit the bacteria. Now we can remember these three drugs from the mnemonic MCQs. Okay, MCQs means macrolides, clindamycin, and quinpristine, and their main mechanism of action is they will inhibit translocation. Next, the fourth way by which we can inhibit protein synthesis and kill the bacteria is by producing misreading. See, normally what happens, we have got codon on the mRNA, and complementary to which anticodon is present on the tRNA. Okay, and it will attach and it will produce protein synthesis, right? But now what we can do is, we have got a group of drug known as aminoglycosides. They will produce misreading of mRNA. Okay, so this misreading of mRNA is done by aminoglycoside. So basically what happens over here is, just suppose we have got AUG as the code on the mRNA at A site, so that tRNA will attach, which contains the complementary codon as UAC, right? No other tRNA can attach. So this will carry one particular amino acid only. When we give amino glycosides, this code is read wrongly, means instead of A in AUC, it can be read as C. So when it is being read C, a wrong tRNA comes. This will contain a different anticodon. So when a wrong tRNA comes, it will carry a wrong amino acid. That means protein which we want to synthesize will not be synthesized, but rather a wrong protein is synthesized, and most of the time this wrong protein is a cidal protein, and it will kill the bacteria. So this is very much important to remember, that aminoglycosides are the only protein synthesis inhibiting drug which are cidal. Like most of the other protein synthesis inhibitors, they are bacteriostatic, but aminoglycosides, they will actually kill the bacteria. Okay, so these are the four mechanisms by which protein synthesis inhibitors can produce inhibition of the bacteria. Now we have discussed the mechanism of action of various protein synthesis inhibitors. Next thing to discuss is that on which ribosome it will act. Like in case 30s ribosome, on the smaller subunit, aminoglycoside and tetracyclines will be attaching, whereas on the larger subunits, the other protein synthesis inhibitors like chloramphenicol, macrolides, clindamycin, and quinpristine will be attached. Okay, now Next, we'll be discussing about the tetracycline antibiotics.